All right, good afternoon. Let's quick start to whatever we are supposed to do today and let's leave because I think most of you are not ready for the class today. That's why you are not coming. So today
Good afternoon, sir.
we are trying to tackle our last lecture for this particular blog and we are going to look at fermentation, which is going to be our last lecture for this blog. And I'm hoping that with this preservation and storage, we've gone far. So today, our main topic is going to be on fermentation. And I think previously we were looking at various processes in which we can handle food and prevent it from going bad. And that has been our focus. So today, fermentation, we are saying that fermentation basically describes a process in which microorganisms change the sensory properties of food to produce an end product that is desirable to the consumer. That is basically what fermentation is. Anytime we are talking about fermentation, we are looking at a situation whereby microorganisms convert sugar into carbon dioxide, into alcohol. Let me put it that way. So anytime we are trying to ferment food, it causes some sensory changes. That is sometimes the flavor and that of the odor, that is the taste of that particular food may also change looking at fermentation. There are a lot of food products in our system that undergoes fermentation, or we use fermentation process to produce them. They might not only be like you are trying to preserve the food, but certain food product itself need fermentation in order to produce the food itself. So that is a specific thing we need to also understand. All right. And we said that it is mostly used in developing countries that lack modern storage facilities. So fermentation as a method of preserving food are mostly used in the developing country. I think Ghana, we are developing. So we may lack a lot of modern facilities that will help us to preserve our food. So fermentation may be another way in which we can explore. What are some of the advantages? We say that it helps to extend the shelf life of a particular food. That means that it helps the food to be kept for a while before it can go bad. And we are also saying that fermentation is often carried out in relatively inexpensive equipment. The processing which that we follow in order to perform fermentation or for fermentation to occur are mostly inexpensive. And that is one of the advantages. They don't demand a lot of money to get their equipment for use. And these are some of the common food in which fermentation is used in their production. Yogurt is one. I think there was a time that I took my time to talk about yogurt and how we use the starter culture or the probiotics to ferment it. We have bread. Fermentation also occurs in bread, whereby we use yeast, which are a type of microorganism that is used to ferment bread. Wine too is another one. Then we can also talk of alcohol production or alcohol beverages. They all undergo what we call fermentation. We can look at soya sauce. The process that they use to also produce it also undergoes some amount of fermentation. Yeah, Anita.
Sir, please, can you give us an example of how the soya sauce and the alcoholic beverages, the fermentation processes, or it will not appear in our exams?
Why are you talking of exams? So if it's not happening in the exam, you don't need to know. Is that what you are saying?
Okay, we need to know
How will I
for
know?
our.
How will I know whether it will come in your exams or not? I'm not your examiner. I beg you.
Okay, so please give me an example of how it's okay. Thank you.
As for example of alcoholic beverage, what we are saying that most of these alcoholic beverages that we've been taking as undergone what we call fermentation. That is what the point is trying to tell us, that most of the alcoholic beverages, and those of us that we have stayed in the village before, it is something that we've been seeing, that you see a palm wine tapper will go and pick all the palm wine from the palm tree, and they will come and keep it inside a barrel and cover it for the next or 2 days before they will even they start to boil it to produce the alcohol itself. So the process in which they pass through is also a form of fermentation. As for soya sauce, I mean, basically the point is telling us that they use fermentation in the production, but soya sauce is not something probably that we can talk in Ghana here, because they are foreign produce that may not be common in our system. But when it comes to yogurt, bread, wine, alcohol production, all these are things that we know them here. Fermented fish, I think we all know Momoni and the rest. They ferment it, they add salt to it and ferment it for some time. So these are something, those ones are common in our setting. And also sausage too undergoes a fermentation process before they are brought into our system. So these are some of the examples of fermented food item that we can talk about. These are microorganisms that are involved in fermentation. I think I have made mention of yeast. We use yeast mostly when we are doing our bread. Mold is also used in some of the fermentation processes. There are bacteria that are also involved in fermentation. And those of you that you have been doing this yogurt issues and stuff, the starter culture and stuff, they are made up of certain type of bacteria. So the bacteria, these are the ones that they've listed them. They have the acetic acid bacteria, you have the lactic acid bacteria. So lactic acid bacteria, then we also have what we call the starter culture. All these are an example of bacteria that are used in fermentation of different type of product. What are some of the benefits of fermentation or fermented food? Some of the benefits, we say that it improves the sensory property of an abundant or unappealing product through development of diversity of flavor, aroma, and texture in food substance. I don't know, those of us that we've enjoyed fermented product before, mostly you realize that when you are taking food that has been fermented, the taste look different. So there is a form of sensory property improvement. They improve the sensory property of it. If you have taken the olden type or the yogurt production that they normally do, those of you that have done the natural yogurt or the fresh yogurt before the addition of sugar, you can clearly realize that the taste is more sour. It is not like the normal yogurt that we see them in the market. So the taste, it is in the form of a sour taste. Okay, that is a sensory property, senses. And sometimes through fermentation, the texture of a particular product can also change. If you have also realized how bread is being produced. See, when bread that does not contain yeast, you see that it is somehow hard, but when you add yeast to it, it proves it and make it more voluminous as compared to those ones that does not contain enough yeast. So that is what we can look at. We can also say that it also helps in the preservation of to preserve harvested or slaughtered products. So fermentation does not only occur in probably either fruit or other product, but you can also get them when it comes to meat products, because some of the meats are fermented. Those of you that have been enjoying the beef and stuff, it also undergoes some kind of fermentation. They keep it for fermentation to occur at a point before it get to our market for us to use. And we say that to increase the nutritional value and digestibility of food. I think a lot of research has also shown that food that has been fermented are easily to digest. They can easily be broken down, but the enzymes in our stomach, which is an added advantage of it. It's also lower the toxicity of it. Toxicity of that particular food product is lowered through the process of fermentation. Toxicity, we are not looking at either the food is going to be a toxic in our system when we consume them. Most of the fermented product have the toxicity properties or materials in it has been lowered by the use of this microorganism that we use to ferment them. And also fermentation also lower the cooking time. So a typical example is our beef. You see, when beef has gone through fermentation process, most of the collagen fiber in it, the one that make the meat more harder, has already been broken down. So cooking it will not take longer period of time as compared to the one that has not gone through this fermentation. There are some basic properties or basic principles that that we use to reduce microbial spoilage in food. And I think at this point that I'm going to talk about, we have spoken about it throughout our lecture, that how do we prevent spoilage? First of all, is to minimize the level of microorganism or microbial contamination in food. And there are various ways that we can use to minimize this microorganisms in food. One of the ways, when you are drying the food, you are trying to remove the water content from the food. What it basically does it. Yes, go ahead. If I need to talk, I'm listening.
So you were talking about it lowers the cooking time. So this condo that we leave it for a day before we make the banku, is it the fermentation process that helps it to cook faster, or can we cook it after the milling of it,
Oh, yeah,
like the
but
same
some
day?
people, some not even the same day. We have people that before they even use their condo, they will let it keep for 3 days before. I remember when we were in the village, that's what our parents do. And one of the key things that they always say, they do that in order to develop a specific taste that they are looking for. I hope you understand.
Yes, sir.
But people
So
also, there were instances that when food is not available, they will just mail it and cook it at the same time, and they will still get the food to you. But what the point that we are trying to establish is the fermentation process itself have effects on the. I think I specifically what I know most is about the meat itself. As for the meat, I can speak about it a lot because fermentation process basically break down the collagen fiber. And we know the collagen fibers are those that make the meat hard. So if those fibers are mostly broken down, it makes the food a little bit tender as compared to the one that has not gone through fermentation. So you can pick a beef that has been fermented and the one food, the a meat itself that has not been fermented, and even look at how the texture, how tender they are, and you'll be able to ascertain that it has effect on it. Deborah.
Madam, sir, please, is also in preparing gakenke.
Okay.
Yeah. So when you ferment it, it makes it easily to cook in
Okay.
preparing gakenke. Yeah. And
All
also
right.
it also brings the nice aroma in the
Yes. Yes.
kinky.
So cooking. Okay. Thank you very much for adding up. So those in the kinky production has also given us an example that with the use of the kinky ferment through the process of fermentation, they are able to reduce the cooking time as compared to those that they. And most of the time, this type of corn dough and the rest, we don't intentionally ferment them, but once we mill them and we leave them for a while, it's basically go through this fermentation process that we are saying. So that is what we can say. So the four main process I want to talk about when it comes to when you're trying to prevent preservation. They are the principles that we follow in order to prevent the food from going bad. The first one is we minimize the level of microbial contamination in food. And I remember when you people were doing the microbiology, you did this microbial growth curve and the rest. There are certain factors that promote the growth of this microorganism. So when you take those factors off, you will limit the amount of microorganisms that can stay in the food and cause the food to go bad. So that is one. You can also inhibit the growth of microflora. That is, you don't want this microorganism to grow in the food. How are you going to inhibit their growth? I think we spoke about it. One of the way was to freezing. When you put food inside the fridge, you've not destroyed them, but rather you have inhibited their growth. That is one thing that we spoke about. We can also kill them. There are other processes that we can also do to kill microorganisms. And one of the ways sterilization and pasteurization. Through the process, some microorganism will die. Even the process of cooking food itself can kill some type of microorganism. Although we know that there are certain microorganisms that even subject them to a higher heat level, they will still be able to survive. But there are other processes such as the sterilization, the pasteurization that we can use to kill this microorganism. So we need to take note of that, and also remove the remaining microbes. Remove the remaining microbes. So if some microorganisms are found in the food, we can also there are processes that you can also undergo them, and so that you can get rid of them. When you get rid of them, what is basically telling you that this microorganism may not be available in the food to cause the food to go bad. One thing that we need to do is microorganisms mostly cause the food to go bad when they are abundant in the food, and when they become too many in the food, they rely on some of the properties in the food to survive. One of them is the nutrients. They rely on the temperature around the food to survive. They rely on water. That is why when we are looking at factors that help promote microorganisms, we made mention of the temperature, the humidity level, the pH, the water activities, and the rest. So all these ones, if they are being taken care of, microorganisms cannot survive. So we are saying that fermentation uses a combination of the first three principles. That is, fermentation should not be expected to sterilize substandard raw product, but rather a high quality product. So what this point is telling you is, because fermentation uses the first three principles, if you want to ferment a product, don't use a substandard product. Don't use a product of low quality. If you want to use a substrate, or you want to ferment a certain substrate, that product should be of high quality. You don't wait for the product to start going bad before you apply fermentation process to it in order to get your fermented product. You may not get the correct one, but once you want to do a fermentation, make sure that the product that you are using, that is what we call the substrate. That is the material you are. So let's say you want to ferment yogurt. The milk powder becomes the substrate, because that is what you are going to put the starter culture in it for fermentation to occur. So when you hear substrate, that is what we mean. And there are also some natural antimicrobial for food preservation. Sometimes, I remember the last time we spoke that most of the time, when after post-harvest, some of the food itself contain live cells that have some properties to fight microorganisms that get into it. So there are natural antimicrobials in food, and these are natural. They can be with the food till probably the food get to some level before their activities may not be relevant to the food. So we say that the spoilage and the poison of food by microorganisms is a problem that is not yet under adequate control. That's why the range of robust preservation techniques. So what we are saying is, when it comes to issue of food spoilage and food poison, which is mostly done by microorganisms, it's a problem that we've not been able to control worldwide. Although we have a wide range of techniques and technology that we are trying to use to control them, still there are issues that is pertaining to that. An example of such technologies or techniques are freezing, sterilization, drying, the use of preservative to meet customer criteria. Food manufacturers are in search for new, more natural alternative that sufficiently assure the safety of their food product. So what we are basically talking about, aside all these techniques that we are using, we still fall short when it comes to preserving and preventing food poisoning. So if we have food product that is having antimicrobial itself, that will be the best, because if microorganisms get to the food and the food possesses some kind of characteristics or quality that will be able to do away with the microorganism, that is the best way that we are looking for, and that is what most of the manufacturers are trying to explore to see how best they can do it. And we see that many plants contain compounds that have some antimicrobial properties, and we collectively call the green chemicals. So the antimicrobial properties that is mostly found in plants are what we call as green chemicals. Green chemicals. So that is a key thing probably I want you to keep in mind. That anytime you hear the green chemicals in plants, they are the substances or they are compounds that are antimicrobial in nature, or that possesses antimicrobial properties that help a particular plant food to fight against microorganisms after post-harvest. And we say that species and herbs, for instance, are well known to inhibit bacteria, yeast, and have traditionally found wide use in food preservation as well as medicinal purposes. So when you pick food such as spices that we've been using and herbs itself, they are known that they can inhibit the bacterial growth, and not only bacteria, and also yeast. They can inhibit their growth. And when yeast or bacteria find themselves in such environment, they become very difficult for them to multiply and cause the food to go bad. So because of that property that they possess, they are being used in some of the preservation processes and also in medicinal purposes. And we are saying that microorganisms also produce a wide range of compounds that influence the growth of other microorganisms present in food. Regarding food preservation, the most important group of single organisms to be considered as a source of biopreservative are lactic acid bacteria. Okay, so what's the point is telling us is when it comes to issue of preserving food, one of the key way that is when you don't want to use synthetic material for preservation, the bio one. Bio means that the natural or the biological way of preserving food, and the typical one that we know is lactic acid bacteria, and they are mostly used and because most of the time in lactic acid is mostly found lactose milk, though the acid that can be also found in sugar. So most of the time when you hear lactic acid, lactose, all these are lactose are sugar that is found in milk. So lactic acid is also an example of a bacteria that can be used in preservation if you don't want to use any form of synthetic way of preserving your food. And we say that they are normally used to produce cheese, sausage, vegetable products. So I think when we started, we made mention that cheese and sausage also undergo some form of fermentation. And fermentation is one of the ways that you can also use to preserve your food. So those of you that have been taking cheese before, the taste will even tell you that it's a fermented product. So, and if you look at sausage, for example, sausage too is a fermented product. They ferment sausage before they are brought into our market for us to consume. So even talking about the cooking time, we all know that sausage, if you even put it in oil for about 2 minutes, you are good to consume them. We say that there are also antioxidants. Anytime you hear the word antioxidant, oxidation is a way that a food can also pass through to go bad. Oxidation. I don't know how with our biochemistry, we did a lot when it comes to oxidation. That is when the food product react to the atmospheric oxygen, and probably it can cause the food to go bad. It can make a food, fatty food to become rancid, can change the color of certain food products. So basically, oxidation can even cause some food nutrient to be lost. Vitamin C and the rest can easily be lost in the food through oxidation. So there are certain food that also can also have antioxidant. It means that those food oxidation cannot occur in such environment. We say that antioxidants are all substances that can protect materials against auto-oxidation, irrespective of mechanism or the action. So any material that you think can protect any food substances from oxidation, you call those one as antioxidant. So let's talk about them. We say rancidity of fat and oil and fatty acids. I don't know whether I should waste time here because I think that this word has been with us since our biochemistry era. So we have fats, oil, and fatty acids are deemed rancid if a significant deterioration of sensory quality is perceived, especially either the aroma or the flavor, but the appearance and that of the texture may also be affected. So those of you that you've been using this fats, this margarine for baking and stuff, when you leave it uncovered for a while, you see oxidation occur. And when oxidation occur, it affects the aroma, the flavor, and sometimes the texture becomes too sleepy that you cannot even use it. Rancidity includes several types of changes, but most often the degradation is due to change in lipid constituents, and this is considered the main feature of rancidification. So most of the time when rancidity occurs in our fats or our lipids, what mostly happens is it causes change in the lipid constituents. Most of the time, I think we spoke about the trans fat, trans and cis, the arrangement. So most of the time, before rancidity this occur, it changes the arrangement of these fatty acids. So if you remember how fat and oil, we have the glycerol backbone and we have 3 fatty acids that has been connected together. So when rancidity occur, it changes the structure. And in chemistry, anytime a particular structure or formula changes, it affects the entire body of the product itself. It can affect the color, it can affect the texture, it can affect the taste of that particular food product that we are talking of. Why are they important? Is antioxidant important? Yes, they are. If you have anything that will prevent oxidation to occur, then it means they are important. We say that the application of antioxidant in human diet for the suppression of free radicals is very important in preservation of chronic diseases, cancer, and aging. So you see, because our body undergoes production of energy, production of new cells, and old cells also die, it produces what you call, yes, we always say that the body undergoes what you call oxidative stress. And through the oxidative stress, it produces what we call the free radical. I think this free radical, if even when we were doing the oxidation, in fact, we spoke about how free radicals are formed at the initiation stage, and the free radical react with each other in the process of causing the fat to become oxidized. So in our body also undergoes oxidative stress, and this oxidative stress produce free radicals. And if these free radicals are not being bound and remove them from our body, they can cause diseases. And a typical example is cancer. Most of the cancerous cells are free radicals that move in our body. Because they are free radicals, they are always available to react with any other substances or compound that they come across. And when care is not taken, they can cause disease. And some of the disease, chronic disease. When we are talking of chronic diseases, I know we all know some of our chronic diseases. Then we can also talk of aging. You may look old, and a cancer is another thing. So vitamin C and vitamin E these are most of the time the natural antioxidant that we can be consuming. That is why we are advised to take a lot of fruit and these vegetables in order to get these vitamins to fight against the free radicals in our system. And we say that the induction process, the stage of very slow oxidation in the beginning of storage of fat containing food, and thus the shelf life may be prolonged by addition of antioxidants. So there are certain food products that in the process of producing it, they add antioxidant to it. And we say that in the process of producing some of this, you have a stage that we call the induction stage. That is the stage in which there is a slow oxidation. So you can have an antioxidant, and I think with our biochemistry slides, we gave a lot of artificial antioxidant that you can use they use in some of these food that they produce, especially any food that contains some amount of oil. Some antioxidants are being used. Now let's talk about nitrate, which is a form of a particular material or chemical that is used to preserve food. Let's talk about them. What is in nitrate? We say that nitrite and nitrate are used in many food as preservative and functional ingredient. They are critical compounds used to cure meat and are known to be a multifunctional food additives. Okay, so they are mostly used when we are trying to preserve meat, and we are also saying that they have some multifunctional food additives. So food additives, it is not always that food additives are bad. Some of the food additive itself can even add nutrient to it. So that is what we are saying. So for example, we say we have sodium nitrite, which play a role in inhibiting the growth of toxins produced by Clostridium botulinum in most of the animal care products. So you can clearly see that when you have nitrate and you add it to your cared products, your cared animal product, what happens is, this most of the time, this materials, this bacteria produces what we call toxins. And I know you came across these toxins, aflatoxins. They are different types. So when they produce the toxins, it is the toxins that will cause the food to go bad. They cause the food to go bad. So most of the time when they are trying to care this product, they try to add this nitrate to it in order that when this Clostridium botulinum produces these toxins in the food, this particular nitrate can cause the food, can just destroy this, these toxins that are produced in the food. So remember we said that in our microbiology, we said that a food can go bad either in the presence of the microorganism itself or the toxins. When they release their toxins in the food, they can also cause the food to also go bad. So what are some of the factors that affect this nitrite that we are trying to use to preserve our food? And I think most of these factors are those that we've been discussing about. That is the effect of pH. We say nitrate is found to be the most inhibitory to bacteria at an acidic environment. So anytime you want to use nitrite in preserving a particular meat, we are saying that nitrite can inhibit most of the bacteria that are acidic loving. Those who want to be in an acidic environment, nitrite can be one of the best substance to use to inhibit their growth. Remember, we say inhibit, not to kill them. It means that when they still find themselves in probably an alkaline pH, they can still function well and cause the food to go bad. And also the effect of oxygen. Nitrite is more inhibitory under anaerobic respiration. So anytime there is too much oxygen, nitrite cannot perform its function better. They always want the absence of oxygen. That is what we call anaerobic respiration. When there is it doesn't involve oxygen. When it's aerobic respiration, it means that oxygen is mostly used. And effect of other food components. So we are saying that temperature, salt, concentration, and initial inculation size also significantly influence the microbial role in nitrite. So anytime you also using nitrite, the component of other food, the presence of other food component can also have effect or the role that nitrite can play in your food. An example is your temperature, and when there is too much salt concentration in the food is no more acidic. So nitrite cannot perform a better role as compared to the one that does not contain that. And also the presence of heat or the effect of heat. We say that the inhibitory effect of nitrite enhanced tenfold after heating due to the formation of an extremely inhibitory substances that is called the Perego effect. You see, the Perego effect is let me put it this way. When you have a food product that contains microorganisms and you are trying to kill the microorganism, and probably you can use heat to kill the microorganism. When you heat the food to some level and the microorganism doesn't die or it doesn't a minute. Let me pick this for a minute. All right. Okay, so let's continue. So what we are saying is that when you have a food product itself and you want to kill certain microorganism in it. Most of the time when this microorganism, when they find themselves in an unfavorable condition, they develop they become spores. They develop a spores. That is, they develop a protective cover that cover them to make sure that their internal arrangement is not being affected by the external environment. That is what we call the spore. So the Perego effect is when this microorganism, when you heat it to 100 degrees Celsius, you will kill them. Then you heat it to 90. Then you cool it down again. Then the next day you heat it to another 90. The microorganism will develop some resistance to heat. And this resistance to heat is what we call the Perego effect. They develop some resistance that they will not be afraid of heat anymore, because the body, you keep on heating them to the point that they think that they are coming to die, and they get to the normal state again. So the body, their system or the internal system will adjust to how heat mostly occurs. So that is what we call perogo effect. So we say that when you are heating it, you have to heat it evenly, heat it to the point that you think the microorganism can die. But when you don't heat it evenly and you get to a point and you stop and you continue to heat it the next time, it will develop what you call the perogo effect. And at the end of the day, these microorganisms will still be within the food that we are talking about. So that is what we need to know. That some microorganisms, the way you process the food itself can make them become heat resistant. And when you get to a point that they are no more afraid of the heat you are subjecting them to, then we say that they have developed what we call the perogo effect. So basically, that is what I can talk about. Then also effect of radiation can also have effect on them. We say that prolonged injection of sodium, effect of radiation. So radiation can also be another issue that can also contribute to whether the nitrate can perform its function. And with radiation, it uses the radiant heat. It uses more of radiant heat and more of gases and the rest. And sometimes it uses light, radiant heat. When you look at the use of microwave uses radiant heat. It causes the food component to vibrate among each other, and when vibration occurs, it produces heat. So that is what you can. Then let's look at some of the factors affecting the efficacy and nitrate. Some of the factors that we've been talking about. We said that prolonged injection of sodium nitrite or sodium nitrate has shown to cause met hemoglobin. Pay attention to this particular point very well. That when you use it too much in our food system, it can cause a problem that is called met hemoglobin. We said that it's a condition in which affects our blood, which causes the production of abnormal hemoglobin, especially in infants. So most of the time, the effect is always with the infant. And we all know that the hemoglobin is having a specific shape. So when the shape is altered in the process of, or in the act of taking too much sodium nitrate or sodium nitrite, this is what is going to happen. And we know that hemoglobin, the shape of the hemoglobin helps us to carry oxygen into our body. You know, hemoglobin play a very key function in our system. It is the duty of the hemoglobin to send oxygen throughout our body. And we know the work of oxygen. Without oxygen, the body cannot break this glucose that we have. The body has digested, or the end product of glucose without oxygen cannot be used to release energy in the form of ATP. So when your hemoglobin shape changes, it affects the amount of oxygen that your body, your red blood cells can, or the hemoglobin that is contained in the red blood cells can send across your body to produce energy that is needed in our system. So we need to. So one of the key effects that you can, when you are asked to talk about one of the key effects of consumption of this nitrite in the process of preservation is that it can cause what we call the met hemoglobin. That is the process whereby your body starts to produce hemoglobin that is having altered shape. Now the shape has changed, and we are seeing that it is very common among children. So we should be mindful when we buy food products. It is our duty to read on it and know how best we can do that. We are also saying that another effect is that it can also cause cancer. It can also induce cancer. So we need to be very much careful. That is why it's our duty to always try to read around this particular food product to know the kind of things that they use in the process of preservation. What about medical or health effects? There are two types of health benefits that may be provided by food additives. That is what we need to. There are two. And we say that those that prevent or reduce the incidence of specific diseases, and those that also provide or enhance nutrition. So as we keep on saying, we said that this nitrate, aside it being a preservative, they also have some functional properties. When we were from our previous slide, that is what we said. They also have certain functional. So we said that some of them can prevent some specific type of diseases. So probably it is something that we can read on it and see the kind of diseases that this nitrate can prevent our body from. And some of them too can also enhance nutrients. And because it contains sodium and the others, these are all nutrients that some of them perform specific function in our system. So we need to understand that. Yeah, Deborah.
Sir, please, can you give me some of the nitrate foods or the additives?
Okay, most of the food that has been preserved with the use of salt contain this particular nutrient that we are talking about. So most of the food that has been used salt to preserve it. So that is what I can say. Sodium contain
Okay,
this
thank
nitrate
you.
that you're talking about. Yes. So we've been able to talk about some health effect and also benefit. So the benefit, we have made mention that they can provide or they can enhance nutrition. They can add some nutrient to your food, and also they can also try to prevent certain type of disease. I would like you to read on the first one. What type of diseases that nitrate can prevent them? I would like you to read on them, and probably you can share it on our platform that we find ourselves. So now pH in food and food preservation. I think when we were doing the preservation itself, we spoke a lot about issue of pH. True or false? Did we talk about pH in food? When we were doing we started this course, I think we've been explaining pH. True or false? Do you remember them?
Sir, please, we've done it.
You've done it. Okay, tell me what the pH means.
Sir, please, it shows
Yes,
the
sir.
alkalinity and acidity of substances.
Continue. What
Also,
is the role of pH in preserving food?
okay, a food that contains a lot of acidity, a microorganism cannot grow in it. Thank you, sir.
Can somebody also say something else? Is that all? Is that all? Hello. Today the usual talkers didn't come to class, so the place is quiet. And Deborah is the only one talking, and I think Anita too is here. Hey guy, you are here and you are not talking. Fika, Cynthia, you are also here. Letitia, you are here. Patricia Mensah. Aside the acidic issue, what again can you say about pH? What again can you say about pH? Hello. Aside the acidic issue, what again can you say about pH? Hey, move me. And I'm going to solve it. You will not mind me. You want to as well. So effect of pH in microorganism and enzymes. So we are saying, I think from our previous class, I think just our sister said, most of these microorganisms or enzymes work best in a specific pH. So when you are talking of digestion, let me cast your mind back to this and probably you may remember. When we are talking of digestion, we say that digestion starts in the mouth, and in the mouth, there's an enzyme called the ptyalin or the salivary amylase, and they convert most of our starch into maltose. But when you swallow your food, within your pancreas, your pancreas also have a specific enzyme that we also call the pancreatic amylase. Our mouth is alkaline in nature, but when the food gets to our stomach, the stomach walls produces what we call the gastric juice. The gastric juice, and we say this gastric juice provides three main things to the stomach. One, it produces what we call there are two enzymes, renin and pepsin. These are proteins enzymes. They act on protein foods. There is also produces what we call HCl, that is the hydrochloric acid, which also produces acidic environment from this renin and pepsin to work. And also these are what is it? This ACL kills the enzymes, that is the ptyalin that accompany the food into your stomach. So what I'm using this scenario to let you know that ptyalin can work best in our mouth because our mouth is acidic in nature. Immediately the food gets into our stomach, this ptyalin cannot work in our stomach. That is what we need to understand. And also the stomach enzyme, the one that we call the renin and the pepsin, because they want acidic environment in order to act. When they are also found in our mouth, they cannot work. So what it's telling you is even within the enzymes, there are enzymes that can perform their work when they find themselves in an acidic environment, and there are also enzymes that can perform their work when they find themselves in a basic environment or an alkaline environment. So that is what you need to just understand when it comes to issue of pH. So we are saying that the pH is a term or it's a symbol of hydrogen concentration. The pH scale ranges from 0 to 14. 7 is neutral. Those before 7 is acidic, and those after 7 is a basic or alkaline. I think we've all learned about this. We said in general, fruit, soft drink, vinegar, and wine have low pH value at which most bacteria would not grow, and this food have good keeping quality. So what we are saying is that a lot of bacteria cannot work within the acidic environment. That is why we say that vinegar and some of the drink have low pH. When you say your pH is low, it means that you are within the acidic medium, and this is where most of the microorganisms cannot thrive. So that is what we are saying. And we say that most meat, seafood, and raw milk have pH value greater than 5.6, which make them more susceptible to bacterial spoilage and possible growth of pathogens. So when your pH is more than 5.6, it means that you are getting closer to the neutral. Because 5.6 is a weak acid. From 0 to around 4 is a strong acid. Once you move beyond 4, you are getting to weak acid. And weak acid because you are getting closer to the neutral. And once you are closer to the neutral, you are weak. I hope you understand. Once you are closer to the neutral, you are weak, you are not strong. The same way that we have weak base and we have strong base. So those that is around 10 and above may be stronger, and then those that is closer, that is around 8, 9. They may be weak because they are closer to the neutral. Once you are closer to the neutral, you are weak. So the point is telling you that most of our meat, most of our seafood, most of our raw milk, their pH is above 5.6. So let's say they are around 6. And when your pH is around 6, you are automatically closer to neutral. And once you are there, it is easy for microorganisms, because we have been saying that most microorganisms can destroy food within the pH of neutral and that of getting to the weak alkaline or the weak base. So this is what we need to know. And we say that vegetables also have a fair pH, higher value, and are more prone to bacterial spoilage. And we know as for what is it? Most of our vegetables, their pH is more higher. They are around the neutral, getting to that of the alkaline. So getting spoiled is not anything that is difficult for this microorganism. We are saying that some of the most important factors affecting pH value of a food is, one, the maturity of the produce. I think the last time we spoke about it, that even if you want your food to be kept long in the shelf, one thing that you need to do, you need to be mindful that the food is well matured before you harvest it. And also variety of cultivar. If the food is having different type of variety, it helps, because when it is only one, the variety is only one, microorganism can specialize in it and destroy it. But when you have different type of variety of a particular food product, and you have added them to produce a particular item, that food, the contamination of that food is quite low as compared to the one that is having only one type. So we are saying that even with pepper, you have green pepper, you have the chili pepper, you have the other types, you put them together, then you produce a certain product. Microorganism trying to cause that particular food to go bad will be difficult than if you have only one type of pepper that it is being used to produce that particular item. And also geographical areas in which the food is also being produced. Okay, a lot of research has shown that plants that are found in the northern part of Ghana, because of the weather issues, because of the hot sun, they are able to develop protective material or compound to protect themselves because of the environment that they find themselves, as compared to an environment whereby there's a lot of rainfall. So when you find yourself in a harsh environment, your body develop a protective mechanism to protect itself, so that it will be able to fight within the environmental factors. So your geographical area in which the food product is being cultivated also have issue, and also the handling and the holding process prior to processing. So after harvesting, before you process the food item itself, those periods, we call it the handling and the holding process. If you are not able to take very good care of the food at that moment, a lot of microorganisms can get there and can change the pH of that particular food and cause the food to go bad. So this is a few things that we need to talk about. Let's look at the issue of pasteurization and sterilization, and probably we can call it a day. I think pasteurization and sterilization are two major heating processes that we use in process of setting food items, which are very common in our environment. So we need to just understand that. We say that with the issue of pasteurization is the process of heat treatment to inactivate enzymes and kill relatively heat-sensitive microorganisms that cause spoilage and minimal changes in food properties such as sensory and nutritional processes. So the issue of pasteurization is very, very important when it comes to preservation of certain food items. And a typical example is milk. Anytime we try to preserve milk, it's undergo what we call pasteurization. And there are different ways that pasteurization is okay. But one thing that we need to know is pasteurization uses a high heat. And because it uses a high heat, most of the microorganisms that are heat sensitive are mostly destroyed, or they are mostly killed. So that is one thing that we need to know. What is the purpose of using pasteurization as a process of, or as a method of preserving a particular type of food? One, to free food from any microorganism that may cause deterioration or endanger the consumer health. So through pasteurization, we will be able to destroy this microorganism or do away with this microorganism in order to protect we the consumers our health. And a food like milk contains a lot of micro because of the protein nature of it. Microorganisms can easily get into this particular food. So if it doesn't go through any system that we can just kill some of these microorganisms and we consume them, they may not be safe for our health. And also pasteurization is one of the most important. Pasteurization is one of the most important steps in preserving, and it is also essential for food safety. So if you want to preserve the food from going bad, we use what we call pasteurization. And pasteurization comes with different temperature and time. Pasteurization uses time and temperature. Sometimes it undergoes probably 200 degrees Celsius for 5 minutes. Sometimes you increase the temperature and then you limit the time, because sometimes these heating methods can also destroy some of the nutrients that is found in the food. So if the time and the temperature is not controlled, probably we will not achieve the desired outcome of pasteurization that we are looking at. We are saying that it greatly improves the product's keeping quality because enzymes are inactivated, microorganisms are killed, and also effectively destroying all disease-producing and other bacteria. So through pasteurization, a lot of these enzymes and microorganisms that causes the food to go bad, they are mostly destroyed or they are killed, because we use a high heat temperature in order to do away with all this one. We say that the severity of heat treatment and the resulting extension of shelf life are determined mostly by the pH of the food. So listen to this point very well. In a low acidic food of pH 4.5, the main purpose is to destroy pathogenic bacteria, whereas below 4.5, the purpose is using heat treatment to destroy spoilage microorganisms. So pay attention to this point very well, that when you have food that the pH is less than 4.5, no, sorry, greater than 4.5, your purpose is to destroy pathogenic microorganisms. Pathogenic means that those that can cause diseases to your system. They can produce pathogens that can cause diseases. When you consume this type of food that contain this pathogen, you may fall sick. And we are also saying that those that are below 4.5, the purpose of using heat treatment is to destroy this microorganism or to make them inactive. So you see, when it is above 4.5, that means the microorganisms has already produced their pathogens already. But when it is below, that means we need to find a way to inactivate the microorganisms so that they will not even be able to produce their pathogen that can cause the food to go bad, or the one that can cause some kind of health issues to our system. Now, these are the types. These are the types of preservation. If you get time, if you can read on it, we have the in-package pasteurization, we have the continuous pasteurization, we have the batch pasteurization, and we have pasteurization prior to packaging. So what we say that the in-package, most of the time, after the food has been packaged already, the food will be inside its package. Then we can apply some heat to it to some point. With specific temperature and specific time. Then also continuous. With that, you change the temperature in a continuous manner. So let's say I want to do pasteurization for about 30 minutes. So the first 5 minutes, I have a specific range of temperature I will give it to it. The second 5 minutes, I will subject it to different type of temperature. The third 5 minutes, I will subject. So it continues till the time limit that is needed for the pasteurization has been exhausted before that method has been used. And also with the batch, that one you portion the food into different batch, then you pass it through the pasteurization process. So probably the first batch of food may go for 10 minutes for maybe probably 190 degrees Celsius, but the second one may not go through the same thing because of the different type of the pH of the food can be a factor. Probably the nutritional component of that particular food can also be a factor. That is why you may use the issue of batch pasteurization, and also pasteurization prior to packaging. Sometimes before the food is even put inside its package itself, there should be some form of pasteurization. So when you pick milk, for example, milk can use different we can have in package pasteurization, because after we have packed itself into the can, we can subject it to heat so that it can do away with most of the heat and most of the bacteria that have found its way into the can. Is another way. Even sterilization we made mention of those things. So keep these three things in mind, that when you are asked to probably mention.
Sorry. The in package, my network wasn't good, so I didn't get it well. So can you please go over for me?
Okay, so what I was trying to say is that there are various means in which a particular food can be pasteurized. Sometimes it can be in its package itself. That is what we mean in package. And we have another one, that is the prior to packaging. That is you pasteurize it before you put it inside its package or the final package that you are going to transport it to your consumers. So with the in package, you put it inside the package itself. Then you pasteurize the food and its package, everything, before the food is being sent to wherever that they are supposed to find itself.
Okay, thank you.
But another heat way of also preserving food is what we call sterilization. I think this one too is another means in which we used to. But one of the key differences that I would like you to go and look at it is one of the key differences is the use of heat. Most of the time, sterilization may use a higher heat as compared to that of pasteurization. You can also try and find out if what I'm saying is true, or if I have not interchanged the two of them. So we say that sterilization is a complete destruction or elimination of viable microorganism or in food to protect or food product being sterilized. So complete. So remember, we said that with the pasteurization, sometimes you may not kill all of them. Sometimes you make some of them inactive. But this one completely, when you subject any food product to sterilization, every heat sensitive microorganism will be destroyed. Every sensitive food microorganism will be destroyed. So that is when you want to do the comparison. These are one of the key things that you can look at. And instead that it destroy yeast, destroy mold, destroy vegetative bacteria, spores, fomites, and also allow food to be stored and distribute at ambient temperature with extended shelf life. So if you remember, when we were looking at this microorganism, the last time I spoke about aerobic microorganism, anaerobic microorganism, and vegetative microorganism. Please, do you remember these things that we said the last time? Hello? Do you remember them? What did we say about them? Aerobic, anaerobic, and the vegetative microorganism. Who is talking? Who remember this thing that we said? No one is talking. So I should continue.
Sir, please, sir.
Rebecca.
If I can remember, the aerobic microorganisms are the one that can produce and grow where there are oxygen. And then the anaerobic ones are the one that they can grow without oxygen. And then when we talk of the vegetative microorganism, they are the ones that do not have spores. So therefore, heat can kill them easily compared to the spores microorganism. For them, they have spores, so killing them with heat is not easy, or it is difficult.
All right, I think you've done well. But with the vegetative, I think one thing that I also stress on
to
it
kill them. So that
yes.
is what I can remember about these three.
All right, that is good. So with the vegetative, one thing that I also stress on it is vegetative microorganism can still survive in both conditions, either the aerobic or anaerobic respiration or environment. They can survive. So that is, I mean, we told you that spores microorganisms, they are those that they are very difficult to eliminate, the spores one, because when anytime a microorganism develops spores, it means that they are trying to develop some protective covers that allow them to live in the environment for a while before probably they can be destroyed. So spores are always difficult to do away with them. So we are seeing that, but when it comes to sterilization, it can even kill the spores. So you see, it means that one is having a higher temperature that can deal with these things as compared to pasteurization and other heat method that we can use to preserve these foods. You said that sterilization procedure involve the using of heat, radiation, chemical or physical removal of cells. So you can use heat, you can use radiation, that is light. You can use chemical, and we can also use physical means that we can just do away with these microorganisms. These are the processes. Sterilization process. The process consists of four distinct stages. The product must be heated from 110 to 120 degrees Celsius to ensure that they are sterilized, or to ensure that the sterilization has occurred. We are saying that the products require few minutes to equilibrate. Few minutes. So remember when I even talking about, I told you that most of the time these heat processes that we use in preserving certain type of food, they are more specific, especially when it comes to the issue of temperature and time. Sometimes when you mix the temperature and the time, you can even cause the product to go bad. You can spoil the product, or you can destroy a lot of nutrients that are found in food. So we say that even though you may need a higher temperature to do that, but the time that you need should be very few. Or when you extend the time, it may be that you are even cooking the food, or you are trying to apply a heat to destroy most of the characteristics of the food. We say that the product must be held for a period of time to gain sterilization value. So when we are doing it, the time that you are using, if you don't allow the time to reach, you may not get the value of the sterilization that you are talking about. Because probably your main focus may be that this heat process. Hello, is your hand up, or Rejoice? Any question?
So please, it's a mistake.
Okay. So what we are saying is, what was I even talking? I was talking about the issue of the timing, that the product must be held for a period of time to gain the sterilization value. So if you don't allow the time to reach and you remove it, or you remove the sterilization process from it, you may not achieve, or you may not be able to kill the number of microorganisms that you want. And also when you make it the time to exceed the limits that you are supposed to move, probably you can also change some of the characteristics of the food product. I would say that the product must be cooled to stop further heating through overcooking. So you see, anytime after sterilization, the food product must be cooled immediately, because you have applied heat. So when you allow it to be in that high temperature for a long time, overcooking can occur. So immediately after the sterilization, you have to remove the source of heat from the food and allow it to cool to a point whereby you can start your packaging and the others. So these are some of the things that you need to know. And methods of sterilization, we say that food sterilization methods are divided into two categories. So we have the thermal and the non-thermal. When you hear the thermal, we are talking about the one that uses heat, and the one that does not use heat is called the non-thermal. So these are two things that I want you to put it in mind, that the two processes for sterilization are the thermal and that of the non-thermal. So when we hear thermal energy, we are talking about heat energy. So we say with the thermal processes, we have two main process that goes through the thermal processes. We have the bulk processing and aseptic processing. So these are a diagram that is depicting the what is it? A diagram that is trying to show you how sterilization occur. So you can see that we have two main types. We have the thermal sterilization, then we have probably the non-thermal sterilization. So look at the structure or the image below, and just understand how the types are. Look at the images below. Look at the images below. Have you seen the processes? So we have sterilization as one, and we have the types, the two main types, the thermal and the non-thermal. And when we come to the thermal, we have the two main types, which is the bulk canning. The bulk canning. Those of you that have been buying can product, these are what mostly happen. Then we have the aseptic sterilization. So let's take our time and see how best we can talk about that. So when we talk of the bulk canning, we said that the bulk canning is basically when you are heating food in a sealed container. So when you look at your hello, maybe it's yes, maybe it's
So you are saying that the thermal processing, we are sterilizing by heating. So then the non-thermal without heating. But the sterilization, we are saying we are applying heat to destroy some microorganisms, or we are inhibiting their growth. So can we still do sterilization without the heat?
all right. Yes, there can be sterilization without the heat. That one, it makes use of what we call a high pressure. I will talk about them. It makes use of what we call high pressure. There's a high pressure that we call HPP, high pressure processing. We use that one too for sterilization, but that one doesn't involve itself. It doesn't involve itself with the use of heating or using a thermal energy. Let me use the word thermal energy. I think that one is more appropriate without the use of thermal energy, without the use of heat energy. So that is what. So let me go through this, and after that, I'll take my time and talk about the non-thermal one for a bit. So we are saying that with the thermal, it uses what is it? I was talking about the bulk canning. That is, we know that we have been canning a lot of food products in our system. We put them in a can or a tin, then probably we preserve them or transport them to the various places that we want to do it. So what happened is, with the issue of bulk canning, we put all the food inside it can, then we seal it. Then after that, we subject it to high temperature, typically from 115 degrees Celsius to 121 degrees Celsius, under the bulk canning. That is what we do. And our goal is to commercially sterilize all the canned food that we are trying to send it, to the market for us to sell them. So with the bulk canning, it's look more like the in-package pasteurization that we did, the bulk canning. But we don't pick the cans and be sterilizing them one after the other. But there will be a surface whereby most of the canned product will be subject there. Then we apply that particular heat from around 115 degrees Celsius to 120 degrees Celsius. So most of our canned fish, we said our canned fish, our tomatoes, and the rest, we use them in doing that. And one of the key advantages is to prolong the shelf life of that particular microorganism. And one of the disadvantages is that it can also possibly destroy the heat sensitive nutrient that is found in the food. And I hope we all know the heat sensitive nutrients. Do you know them? The food nutrients that are heat sensitive, do you know them? Do you know them? So vitamin C is typical example. A typical example is vitamin C. It can easily be destroyed by heat. So we need to just understand that. So that is how the bulk canning is done. So you are just trying to sterilize the can. The food products in the can itself, and also the can, so that any microorganism that is found in it will be destroyed. Then also the aseptic sterilization. It is more like the bulk canning, but here what happened is, with the aseptic, we sterilize the can itself. That is the thing that we are going to put the food inside, and also the food itself. We don't do it together as in the case of the bulk canning. So that is the only difference. That is the only difference. With the bulk canning, you do everything together, the food itself and the can. You sterilize it, you subject it to heat at the same time. But with the aseptic sterilization, you separate the two. Probably you can sterilize the can itself before you place the food inside, and later on you try to sterilize the food itself. So that one also uses temperature around 135 to 150 degrees Celsius for a few seconds, very short seconds. You don't keep it for a long period of time, because if you do so, you may overcook the food product that you are working. And we are saying that we mostly use them in a liquid food, such as our juices and the soups that have been packaged. So nowadays we have these palm nuts that have been put already processed, that when you just go and buy them, then you start using it. Most of the liquid ones, we do that. One of the advantages, we try to maintain the flavor and the color, and the disadvantage is that you need an advanced equipment before you can perform this particular type of sterilization. So with the non-thermal processing, we said that this involves sterilization, reducing microbial load without the use of heat. It helps to preserve fresh quality, nutrient, and flavor of food whilst achieving microbial safety. So one, we can use high temperature processing, the one that I made mention, that's the HPP. We can also use pulse electric field, PEF, which uses a short, robust, high voltage. So we can use electricity voltage to do that particular kind of sterilization. So you see, when I apply electricity to a particular food product, I want a high voltage to just hit it, so that any microorganism in it can die. With that one, I'm not using heat. That is why we are saying that with a non-thermal, it might not use heat. When I touch when I hold the electricity voltage, it will shock me very well. I hope you understand. And the same thing, when I pour a very hot substance on myself, it may also hurt me. But you can see that with the electricity, it doesn't involve the use of heat. But with the thermal, or when I'm pouring any hot substance on myself, that one involves a lot of heat. So we are saying that there can be a sterilization without the use of thermal energy. That is the use of heat energy. And I've given you can also do what we call ultraviolet rays. We can use ultraviolet rays to cause sterilization to occur, and that one too, it doesn't make use of heat, the ultraviolet rays. So that is one thing that we can also, and ultraviolet is one of the ways that we can also use to destroy a lot of microorganisms in our system. So just have those things in mind. I think I've given you some few processes that we can use to do the non-thermal processing. I think we just finished talking about canning, so it's right for us to just look at it tomorrow. We said that canned food may be described as full moisture, ambient temperature, and stable food product because it has already gone through certain type of sterilization whereby these microorganisms has been destroyed, and most of the time, canned food we allow it to be in the form of moisture. So sometimes anytime you buy canned food, you see that some liquid around it, either being oil or even if it's tomatoes, you can see that it is in the form of the moisture content is somehow high. They always want an ambient temperature, so they always tell you to keep it at a cool, dry place so that the temperature will be good for the product. And also, we're saying that prior to the closure, the product is heated, expanded, and dissolved gases get excluded or driven out. So, in the process of, especially when you are using the aseptic sterilization, okay, you heat the food to the point that all gases in it will be, when you heat gases, most of the gases will expand and they will driven out. And the more you apply heat to a particular gas, it will expand. It will expand and they will live wherever they are, because some of the gases can be a problem or can cause the food to spoil. So they try to get away with some of the gases, especially those that are favorable for microorganisms. And which of the gases is favorable for microorganisms? We spoke about it some time ago here in this particular class. Which of the gas is favorable for microorganisms? Which of them? Which of the gas is favorable for microorganisms? Oxygen. I think we spoke about it. Oxygen. Oxygen is very favorable for these microbes. When microorganisms get to environment where the oxygens are good, they are okay. But too much carbon dioxide, it doesn't help their growth. So we need to just know that. And we are saying that the quality of the canned food, the purpose of heat sterilization is to extend the shelf life of food while minimizing the changes in nutritive value and eating quality. And we all know that once we are working with heat, if care is not taken, we may end up destroying all the microbes. We may end up destroying all the microbes and that of the heat-sensitive nutrients that are found in it. So let's look at a few effects of this heating process on the nutritional component. We are saying that dry heat, when there is a dry heat on some of the food, there is a loss of total isolants into canning liquor, dilution, and dehydration. So when you apply this dry heat to most of this canned food, there is total when you apply dry, let me come again. The effect of heat processing on major nutritional component. First of all, dry heat. Then the second one is protein. The third one is carbohydrates. Then our dietary fiber. So first of all, let's look at dry heat. So when you apply dry heat in most of our food, what it does is it causes dehydration. That is the food, most of the food or the liquor. Most of the time, this canned food and the liquor, they contain a lot of water. So what happens is when you apply dry heat, it loses the water content, especially when you apply it for a long period of time. It will turn most of the water content into vapor, and when it gets to the vapor stage, most of this vapor will be lost through what we call vaporization and the rest. And also protein loses its essential amino acid and lost digestibility. So anytime you apply this high level of heat to most of this protein food, especially the meat content, they loses some of the essential amino acids. And remember the essential amino acids that we spoke about. The essential amino acids, we discussed them in our is it which causes that we read those things, and they lost their digestibility. It make digestion more difficult when you apply a lot of heat to this protein food. And also carbohydrates. We all know that when you apply heat to carbohydrates, gelatinization occur. Okay, gelatinization occur and it increases digestibility. And we all know, as for carbohydrate, when you apply heat, especially gelatinization is more of when you apply wet heat to it, it becomes gelatinized. Okay, the starch, it will swell and burst, and you see that some kind of gel will be coming out. But when you apply this dry heat, let's use sugar for example. When you apply dry heat to sugar, it will melt, it will tend to caramelize, and it will become syrup. And through further heating, it will get to carbon, and that is where you see that it will give you some bad smell and stuff. So these are some of the heat on effect of heat on some of the food products. And also dietary fiber. We said that dietary fiber lipids. So we are going to talk about lipids. We said that conversion of cis fatty acids to trans. Remember in our biochemistry, we said that most of our lipids, their structure in nature is always cis. The arrangement is always cis. So when you apply in nature, or you apply heat to it, it transforms it into what we call trans. That is why trans fats are not healthy for us. So one of the major problems we are facing is people who use oil to fry foods. You know, those who fry the yam, those who fry fish and the rest. They will fry the yam today. They will when they are done, they will pour the oil down. Then tomorrow they will use it. One oil, they can use it for the whole week. And at the end of the day, it will tend to trans fats. That is why we have what we call trans fatty acids. So when you buy any food product that has been preserved, when you read on it, you will see if it contains trans fats. So people who are having issue to do with cardiovascular diseases, the heart, and the rest, we are mindful that they shouldn't take this trans fat, because this trans fat can cause a lot of problems to their system. Not only them, we are all prone to that. Trans fat are not good for our system. The natural fat that our body needs is the one that is cis, and the cis is natural. They are natural. All fats or all lipids occur in a cis form, but through the process of processing a particular food product, we may change the cis to trans, which is not healthy for our system. Then also water-soluble vitamins. I think I have made mention of, we know that the vitamin C and the vitamin B are the water-soluble vitamin. And when you subject them to heat, we lose them. They may leach into the the water that you are cooking in it, or the oil. So we have been advised that sometimes this water-soluble vitamin, when you want to use them to cook your food, the liquid that you use to cook them, it's better you use the same liquid in the process of cooking the food. So let's say some of the B vitamin may be found in some of our leafy vegetable that we use. Probably you boil the leafy vegetable and you want to blend it and add it to your soup. You can use the same liquid that you use to boil it and use it to blend it and add it, so that you can still maintain some level of the nutrient, instead of just throwing it away. Because most of the time when you throw it away, it becomes a problem. All right. And we say that we also have fat-soluble vitamin, and we all know them. They are DEC. The fat-soluble vitamins are the vitamin A, D, E, and K. They are DEC. We say mainly heat-stable. There are losses due to oxidation of lipid and leaching. So we know most of the time this fat-soluble vitamin, they are more stable as compared to the water-soluble vitamins. But when you continue to heat them, you may expose them to oxidation. And when oxidation occurs, we all know what will happen at the end of the day. And we also have minerals. Say that there are possibly increase in calcium and sodium mineral by uptake from liquor. So when there are certain type of mineral that when you apply heat to them, their absorption or their uptake become more easier as compared to those that have not gone through that. What about cooking and frying? Cooking methods. You say heat maybe where we have gotten to, they are just things that we've done them before. That is why I'm just rushing through. So once you have the slides, you read them, you'll be fine. Cooking method, we say heat may be applied in a number of ways. It may be dry or moist heat, or it may be applied by means of fat or infrared radiation. So these are the type of heat. So we have dry heat. If you look at our cooking method, dry heat, we have baking, we have roasting, then we have grilling. These are all dry heat. They are dry heat because probably we don't put the food inside a particular liquid before we prepare them. Then we have the moist heat. Moist heat, mostly the food come in contact with liquid. Then we have the boiling, we have the steaming, we have the poaching, then we have the stewing. So the only thing here is probably the steaming. With the steaming, we don't allow the food get in contact with the liquid itself, but rather the vapor. We put the food in a colander, then we subject it to the boiling water. Then the vapor will pass through it, and the food will be cooked. So all those one form part of the the moist heat. And we said that when it comes to issue of frying, we make use of fat in cooking the food. Then we also have microwave cooking, whereby we are all aware of them. Then we have the slow pressure cooking. So there are pressure cookers. There are pressure cookers that you put your food in, you give it to electricity, then you plug it in. That one can cook the food very fast within a short period of time. And it's a way of also making a food tender. So what are the effects of these different cooking methods on our nutrients? We said that fat, when fat are heated, they melt. If they contain water, the water will be driven off and turn into vapor. Excessive heat can cause the fat to burn and produce rancid smell. So sometimes when you are preparing food at home, then you want to pour some oil on top of the stew or whatever that you are preparing. Sometimes we cut onions and put them inside and heat them. So sometimes when you heat it and the heating get to some level of temperature, you realize that it's giving you some bad smell. Because have you all noticed that? Have you all noticed that? To be giving you bad smell, it means that the heat has gotten to some level that all the fat, we are destroying the fat. Immediately you put fat on fire, all the water content in it will turn into vapor. Because we know that as for water, immediately you get to 100 degrees Celsius, all the water will turn into vapor and they will be driven off from it. Now carbohydrates, when carbohydrate is exposed to dry heat, carbohydrates are broken down and darken in color. I think this was what I was using the sugar for example. I will say that example, sucrose brown or caramelization and finally chars and become dark. It becomes too black when you continue to do it. So those of you that you have been doing treacle at home, the one we use for the gingerbread, it goes through this particular process that I'm talking about. But when you use a moist heat, what happens is the starch granule absorb water and they burst, they swell and burst, and gelatinization occurs. So if you are cooking yam or cassava, this is what you observe. When you finish and you are trying to drain the stock, you see that it is very thick in nature. It is telling you how moist heat has caused gelatinization to occur. So the starch will absorb the heat first. After absorbing all the water, it will swell. Then when it swells, it starts to burst, and when it bursts, this is the gelatinized that will start to come out. We are also saying that the polysaccharide starch, and we all know polysaccharide, they are the complex carbohydrate, the polysaccharide, they are the complex carbohydrate, and they are known as the cellulose, the peptin are important constituents of fruit and vegetable. On cooking, insoluble cellulose changes a little except to soften as it gelatinizes and peptin become more susceptible to dissolve. So we know that when you pick cabbage, for example, they are all form of cellulose. When you pick orange, for example, when we are using orange and stuff to prepare jam and marmalade, we all know what happened. the fiber in it become very soft. It becomes very soft. That is what the point is telling us. It said allow the cell to separate from the main fruit and vegetable and make it easier to eat. So anytime you apply this heat to this type of cellulose, one, digestion becomes very easy. Two, it becomes very soft in nature. It breaks down all those fibers in it and make it easy for us to consume. And we say that fruit with high pectin content, such as apple, becomes soft and pulpy on cooking. We all know that as for apple, a little amount of heat that you apply to it, it becomes very, 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 very soft. And it becomes easier for you to consume them. As for protein, I think these are all old knowledge that we have it already. We said that protein coagulates, and I think you did this in interscience. Protein coagulates when heated. Excessive or prolonged heating denatures the protein. It will denature. So too much heating of protein is not something that is desirable. But in certain aspects, they may be good. If you want to use egg in preparing salad, at least you need to apply heat for them to coagulate before you can get the shape you are looking for to perform your food itself. So those of you that you have been smoking meat a lot, you see, look at the quantity of meat you put it on the smoking rag. And at the end of the day, it will denature. The nature will just change. And when we were talking about enzyme too, we spoke a lot about denaturing and stuff. And once proteins start to denature, they are no more functional. Anytime protein denatures, their functionality is always off. So we need to understand that. Then with the issue of minerals, we say that heat does not merely affect mineral elements found in food because they are stable, because they do not break down at the temperature used in cooking. So when I was talking about, I told you that it doesn't mostly lose, but sometimes it rather enhance the uptake of some of the minerals. Yes.
Please, if I understood you, does it mean that the meat that we have been smoking to reduce the fat is not good?
Come again. I think there's noise there.
I'm saying that is it not good for us to smoke meat to reduce the fat? Is it not
to reduce
good?
what?
The fat from it. Like fish, when you smoke the fish, you said it denatures the fish, the meat or whatever. So is it not good for us to smoke fish?
No, they are good. What we are talking about is not the fish entirely. We are talking about the protein content of it. We are saying that anytime you apply heat to the protein changes in shape. I hope you understand. It changes, it denatures. So pick egg, for example. When you break egg and you fry it, look at the form in which the egg is. And when you finish frying, you look at the shape that you get. It has denatured. It has automatically denatured. I hope you understand. And we use this concept when we were doing enzymes. I think if you remember when we were talking about enzymes, we told you that most enzymes are made up of proteins. And immediately you apply a little heat to it, the shape changes. And when the shape changes, it cannot perform the function that they are supposed to perform. But here, what we are trying to tell you that anytime you apply heat to most of the protein products, it will denature. But we are not saying that it is not good, no. Because if you don't even denature it, how are we going to consume them as human beings? We need to always apply heat before we can eat them. I hope you are with me.
Yes, sir. Thank you, sir.
You are welcome. As for vitamins, I don't think I have to talk much. Well, we all know that vitamins, especially the water-soluble ones, can easily be destroyed by heat. So that one day, I don't think I have to waste much time on that. Now let's look at freezing. Freezing as another way of preserving food. We said that freezing is the activity of keeping food in a low temperature to inhibit activity of microorganisms. I think previously we have learned a lot of issues with regard to freezing, that when the food gets to 0 degrees Celsius, it starts to turn into ice. Then it will from 0 to 10 degrees Celsius, everything will turn into ice and it can be kept for a while. And I told you that when you are freezing food, it doesn't mean that you have basically killed all microorganisms, but rather you have made them inactive. You have inhibited their activity. So when they get a favorable environment, they can still thrive within your food to cause their food to go bad. So we need to do that. We said that freezing causes changes the physical state of substances by changing water into ice through the removal of energy in the form of cooling below freezing point. And I think we are all aware that anytime you are trying to freeze a particular food, you change the physical state of it. Especially the food is always changed from the liquid state into that of the solid state. Sometimes your fish can turn to a solid that if you don't put it for a long period of time, you cannot even use it. So that is what we need to understand. We say that the freezing of food slows down but does not stop the physiological and biochemical reaction and the growth of microorganisms that cause the food to go bad. And I think I've made mention of that. Whenever you are freezing a particular food product, you have not killed anything. You have not changed anything. The only thing is you are slowing down the activity of this microorganism. So we need to be mindful of how we handle freezing foods. We say that fast freezing produces better quality food than the low freezing because rapid freezing results in less cell dehydration, mechanical damage to cell wall, and that of shrinking. So what you are saying is when you have a system that can freeze the food very fast, it is better than trying to freeze the food in a slow process. Well, what we are saying is when you use a slow process to freeze food, it causes dehydration. The cells in the food will lose a lot of water. And when they lose water, it causes mechanical damage. When there's no water, it will also shrink. It will become very small. It will shrink, like the size will become very small, especially when you finish freezing it and you try to thaw it and all the liquid leave, it becomes very tall. But when you put it inside and it is able to freeze within a very short time, then probably you can keep most of the quality of these food products compared to when you are trying to slow the freezing process. So one thing you need to know is when you are using a slow freezing process to freeze food, you are trying to basically cause dehydration. And anytime food product loses water, you know, when your body loses water, you know how shrinkage you become. You will even grow lean as human beings because your cells has lost water. So we need to be mindful of that. And we say that the law of quality frozen food depends primarily on one, the storage temperature, the quality of a frozen food, one, the temperature is one, two, the length of storage time, and three, the thawing process. So let me take my few time to talk here. The storage temperature. When you put a food inside a refrigerator, you put the temperature around, let's say 5 degrees, you freeze it around 5 degrees Celsius. Probably it may not be able to freeze quickly as compared to when you are freezing it around negative. You know, anytime you are freezing and you get to the negative, freezing become proper as compared to when you are within the positive figures. Because we all know that freezing starts when the temperature is around 0, getting to negative. So if your temperature is not good, the quality of the frozen food may be lost. And also the length of storage. When you store a food for a long period of time, you lose the quality. So the last time we were doing some of the food, they gave us specific days and specific weeks that they are supposed to be in a fridge or in a refrigerator. So we should be mindful of that. And also the thawing process. Anytime you are trying to remove food from the freezer and you want to use it, our mistake is we wait when it is 30 minutes for us to use the food, and we will remove them from the freezer. That is not the best. If you want to use your meat in the next day, you remove them overnight and put it inside the fridge. You move it from the freezer and put it inside the fridge. Then the temperature will not be too rapid. When the temperature is too rapid, you loses some of the quality of the food product itself. Remove it from the freezer, keep it in the fridge, and allow it to thaw overnight. By the time you woke up in the morning and all the ice has gone, then probably you can use it. But when you remove it and you put it outside the fridge, the temperature reduction becomes too rapid. And when it does that, it doesn't help in the process. This microorganism, it may become very active within a short period of time, and they may cause the food to lose some of the its quality. So we need to be mindful that if you want to achieve good quality of your food products, you should think about the storage temperature, the length of storage, and that of the thawing process. So we need to be very, very mindful of that. We are saying that microorganisms differ in their sensitivity to freezing, as some could survive or they could be completely destroyed. So some of the microorganisms, when they are in the fridge, they cannot survive anymore. They can be destroyed. Some of them too, they can be with your food for quite a long period of time. So when they still get their favorable environment, they can still find a way and cause your food to go bad. So we say, for example, in fermented food such as yogurt, frozen storage increases viability of beneficial culture incorporated because of their potential health benefit and control of this microorganism. So this type of food such as yogurt, yogurt itself, the microorganisms that we use for the fermentation, they are beneficial to our system, we human beings. A lot of research has shown that this microorganism that we use them in producing yogurt can even fight a disease called white, the one that is mostly common in you women. So even when women get such diseases, there's a recommendation that they can consume this type of yogurt, and most of the time the fresh yogurt, because the one that a lot of things have been added and probably the culture may not be active at that time. So they are very good, and because they are very good, when you are trying to freeze them, it is important to freeze them in an appropriate temperature so that when you consume them as human beings, you may get the benefit that they are supposed to give it to us. What is the effect of food when you are freezing them, or when you are effect of freezing on certain type of food? We all know that when you freeze, all the liquid in the food will become ice. It means that you have dehydrated the food. It will just turn into solid state. So it is something that we should know. And it's the loss in weight of the frozen food. So you can see that because it has lost water, the weight may not, it will lose weight, or the weight might not be the same thing like you have basically the time you are freezing it. And also protein may also denature. So any protein food, you put them inside the fridge, you change the nature will change. It will denature completely. The shape will denature. Pick something, any solid protein, when you put it inside the fridge, you have also denatured it. You have turned from the solid into liquid, from liquid to solid. Example is milk. You have denatured it. And solubility changes. Once it has turned into ice, it can no more be soluble in water. It cannot dissolve unless you remove it and probably allow it to thaw before the solubility, you may get them. And it also affect the texture of frozen food. So sometimes a lot of people, one of the reasons why people don't like food that has been kept in the fridge for a long period of time is sometimes the texture, when you put it in the fridge for a long period of time, you may not get it like the way or the process that you put it. And we're also saying that for some seafood, the development of rancid flavor and coastal failure during that of a freezing process. So some of the seafoods may become rancid. Their flavor may become rancid. That is the foul flavor that they normally come. When it is being kept in the fridge for a long period of time, they may develop that particular flavor. I think we are true. Then we say that oxygen causes oxidation. Oxygen causes oxidative rancidity in food, which with unsaturated lipids, which loss in color and also development of off flavor. This is also linked to our oxidative process that we did. So we are saying that anytime you have unsaturated fatty acids, and we know that unsaturated fatty acids are those that contain the double bond and that of the triple bond, and those are those that are susceptible to that of oxidation, because once they contain double bond, some of the hydrogen has been lost. But those which are saturated, the saturated ones they don't normally undergo what we call oxidation, and it can also cause off flavor. Anytime oxidation occurs, there is off flavor. The flavor that comes off are very, 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 very bad. And we say that this is a high during fish storage, and because we know that fish contain, do you remember when we were doing biochemistry, we said fish contain linoleic and linoleic acid. That is omega-3 and omega-9, omega-3 and omega-6 fatty acids, if you remember. So that is why we are saying that it's very high in when it comes to fish. And so when you put them in the fridge for a long period of time, this is what you are also going to get. We say that poultry fat become rancid after long storage. Poultry fat, they become rancid after you have stored them for a long period of time. So today, after the lecture, go to your fridge. Any meat that you have kept it for a long period of time, I think we should remove them and start using them before we lose most of their property. And we say in fruits and vegetables, most important color changes related to some biochemical and physiological mechanism. And these are changes in natural pigments of vegetable tissue. We all know that most of these vegetable and these vegetables and plant produce, they come with its own color. So when they come with its own color. So when you keep it in the fridge for a long period of time, the pigment may be lost. And sometimes when the color even loses, it doesn't even become attractive again. And two, development of enzymatic browning. So sometimes because you have kept them inside the fridge for a long time, you see that the color is even changing. And those changing of color, they are enzymatic browning because they involve enzyme. Those that they don't involve enzyme are those that we call them the non-enzymatic browning, the caramelization, the Maillard reaction, and the others. Those are the non-enzymatic browning. And also breakdown of cellular chloroplast. A green color vegetable is lost by chlorophyll degradation during freezing and frozen storage. So we know that most of these green vegetables contain what we call the chloroplast. You know, it has a specific pigment called chloroplast. This chloroplast help it to trap the energy from the sunlight, which is called the chlorophyll. So when you keep it inside the fridge for a long time, those ones, it will lose the pigment. And when the pigment is lost, it affects the color and the chlorophyll that is contained. So we should know that vegetables should be kept in the fridge for a short period of time, then probably we use them. Then also irradiation. They are saying that as for irradiation, that is where microwave uses radiation process to heat your food for you. So we say the high dose of irradiation can produce or cause changes in chemical composition and taste of fish and seafood. Irradiation can be combined with freezing to retard spoilage in food. So one thing that we are saying that through the process of radiation and freezing, we can use it to prevent food from going bad. That is what the point is trying to tell you, that we can combine irradiation and that of a freezing process to retard this microorganism that causes food to go bad. So we can irradiate the food first, then after that we put them in the fridge. And most of the time we may lack the machines that we may take them to cause this radiation to occur. So we are saying that microwaves are electromagnetic radio waves that are within the frequency beyond 300 megahertz, 300 to 3,000. I think there is a mistake, 300 to 3,000 megahertz. So we need to understand that microwave also uses what we call the electromagnetic waves. Electromagnetic, that is electric and magnetic has come together. So when the light of the microwave touches your food, because it is made up of light and magnet, it causes vibration within the food. So sometimes when microwave is even on, some of you can even hear the way the vibration goes on. It causes vibration within the food. So as the food is vibrating and hitting one each other, they produce some heat, and this heat is the one that mostly cook the food or heats the food for us to enjoy. So these are some of the advantages of using microwave to cook. And I think when we are within our home, we can easily read this and we will be fine. We said that cooking takes place thoroughly and whole volume of food internally, uniformly and rapidly as heat penetrates inside the food material, significantly reduce the processing time and energy. So we all know that when you are using microwave, it uses a short period of time to heat your food as compared to when you are using other methods to prepare the food. And because with microwave there, because it is using some electromagnetic waves, it can penetrate the food at the same time. That is why rapid or uniform cooking can occur. When you give it a time, you pick the food and you see that every part of the food is well cooked when you are using or well heated when you are using microwave as compared to the other. And we say that since heat transfer is fast, nutrients and vitamins content are well, they are preserved and they may not be lost. That is what the point is trying to tell you, that because it takes place within a very short period of time, those heat sensitive nutrients may still be maintained as compared to any other food cooking method that take a longer period of time before it can be cooked. And we are also saying that they are high heating efficiency. Yes, it's very, very, very efficient. Very, very, very efficient. As for when it comes to microwave, we don't waste a lot of energy. Most of the energy are channeled in the process of heating the food. And it is suitable for heat sensitive fluid. Heat sensitive. Most of the food that are heat sensitive, that when you apply other heat to it, you may lose the liquid texture of it. You can use microwave to probably do it and it will be fine. And ultra fast pressurization minimize nutrient, color, and flavor loss. So we may not lose a lot of color, flavor, and that of nutrient when you are using microwave to prepare your food. So we are done. I think we are done for the day. Can on your microphone and you have any question, you ask me, then I will prompt you. Yeah, Sarah.
Thank you very much, sir. Please, I wanted to suggest that the class.
Sarah, your network again today too. And I can't hear you. Is there any other question? I can't hear Sarah, so if anyone is having an issue. I can see she's talking, but I can't hear her. Sarah, your network is not good, so you can type your message at the comment section so that I can read it to the whole class. So I can't hear you. Is there any
Please,
other question?
yesterday you were saying when we said we're confused, you were saying you would try and make 1 hour for us within the week. So that's what I'm suggesting. If tomorrow will be okay with you, most of us will
Tomorrow
be home
Monday.
tomorrow. Yes,
What
most
time?
of us will be home.
What time?
But any time that is okay with you, we will be
Unless
home. We are on midterm.
unless you people are on midterm, unless in the evening, because tomorrow I will go to
Yes,
school. And probably we can, it's 7 o'clock will be fine. You think 7 o'clock will be okay?
okay,
With
sir.
7 o'clock. No, let's discuss as a class so that we can just have conclusion on. Well, 7 o'clock will be okay. And the unfortunate aspect is there are other people who are not here. The Maoli people, they are not here because I don't take them through this course. So let's discuss it on the platform. 7 o'clock. If it is fine, then tomorrow I can meet you 7 o'clock. Then we can conclude on that.
Okay,
If are
sir.
you okay?
Thank you.
The rest of the people, are you okay? Are you okay with the 7 o'clock? If you think it's not going to be favorable, you can let me know. Hey, no one is talking.
Oh, sir, it's okay.
Okay, so then probably let's meet tomorrow, 7 o'clock in the evening, then we can conclude on our calculation. I will spend just 1 hour. So the one that we were solving yesterday, try your best and go through. I will see if I can get some question myself and give it to you and see if you can try your hand on it. So if you don't have any other question, then let's call it a day. Okay, then have a nice evening. Enjoy the rest of the weekends and your midterm.
Thank you, sir.